noticed is that it's actually even more complex than this, right? So the 20,000 genes actually encode more like 400,000 or 500,000 proteins. And if you take the gene expression panel or profile before or after any one inflammatory cytokine, it will look very different early in the disease, late. And if you take a protocol and share it, you still can have data that's very unrelatable between labs. It's often hard to recapitulate others' work. From sort of an organizational standpoint, how do you think about organizing all these amazing scientists to make that work relatable? What technologies are you using to try and bridge these data so that you get the most out of it?
I'll answer in 3 parts. The first part is that these techniques actually reduce the amount of these types of problems. And the reason for that is that often the lack of agreement between studies was that the proportions of the cells were a little different. And once you averaged, that led to radically different results. When you're looking at the individual cell level, it is easier to see the similarities in things that otherwise would look distinct. This was our assumption. We now actually know it's true. So when I show the slide, which I can no longer show, I apparently can, thanks to the kind AV people. Let me try. When I show this slide, each of these blue boxes is made of efforts of multiple labs in multiple institutes in multiple countries, often analyzing individuals of great diversity. And what we have learned from this exercise already inside what we call our biological networks, which bring together all the scientists interested in one organ, is actually the remarkable agreement between their findings. We expected a much worse outcome. The reason for that is partly what I already said, but partly also our computational algorithms. So computational algorithms, if they actually know that these 2 samples should have been the same, they can use, or roughly the same, they can use this information in order to relate the information to each other. And I'll do this by analogy. These are actually similar algorithms to the ones that Google Translate uses for you. It takes one language, and it takes a second language. And it knows, here's a whole set of documents written in one language, here's a whole set of documents written in another language, but at the end of the day, they're actually the same documents, right? It's translations and so on. Match them together. Find that this word means that word by putting them somewhere together with each other. And they actually put them into images just like the ones I showed you for cells. So that is actually what we do. And we have learned from this that our ability to understand biology really just like leaps over this huge hurdle of variability that we've had in the past, mostly because of the great quality of the data, the great resolution of the data. Thank you.